Thank you for attending on late on a Friday afternoon. I've recently joined Solvay. I've been there for only about a month, and so in days, everything that you see here will be in a GitHub repository. I'm in that position where I have to convince another set of managers that while the work I'm doing is interesting enough to be presented at a meeting, there's not a competitor on the face of the earth or perhaps in the universe that can make any use of my work, and so it's being public is an okay thing. So, however, they did say that I can share my work with friends. So friends, at the end of the meeting, drop me an email and I'll just send you the slides in advance of their getting up on GitHub, because they're the minions are seeing just what it is that I'm doing. That wouldn't be bad. I like that. That works out. So, so that having been said, big room. I'm hoping this is more of a conversation than a presentation. So if you have questions, please ask them. I'm going to start with a set of slides to try to give you context for the work that I'm doing here, and then go into a few R notebooks and a Jupyter notebook. And I'm happy to share those. And those are the real code. So what there are two things I embrace. So I sent in the abstract, and it came back saying that this would be, well, data for the common good, and I hope I'll be able to present a little bit of that with the anti-malarial work, but also that it's meant for beginners, such as, but more to the point that it's meant to be a workshop. So I'm hoping that as you have questions, ask them, and I'll do what I can to answer them or avoid them. So that having been said, if you ask really hard questions, I have some licorice. So if you feel a question bubbling up, give me like take 5 or 6 of these and just start chewing on them, and then I think we'll be all set. So, but those are for everybody, right? Oh, by the audience, I'm not proud. And now, there we go. I was at Syngenta, an agrochemical company, an international company. And one of the things that amazed me is they always had beginning meeting questions. Not, icebreakers are just something that was supposed to bring everybody together. And I kid you not, we used to have to sit in a circle while we had these questions. And I was waiting for the talking stick to go around. But so they once had me, it fell to me to come up with one of the questions. So here it is. And, well, okay, the right answer gets some licorice. Oh. D, oh yeah, D, first letter of the alphabet. This is a bright group. So, and clearly after this, I wasn't ever asked to do a beginning meeting question again. The goal of this work is to predict new data using models. So hopefully in this next little bit of time, I'll describe how I've built models for its anti-malarials. And you'll see some of those structures in a little bit. And the idea of machine learning is a lot of compounds have been screened. So what can we learn from those screens to predict the next set of compounds? And where the rubber meets the road is I was working with MMV, Medicines for Malaria Ventures, and they are a paper company, if you will. They're theoretical. They do not have labs. When I first went to visit them, I marveled at the fact that they were in an office building and there are no fume hoods. And so they're essentially middlemen and middlewomen. And they contract with laboratories to make or purchase compounds that then they contract other labs to test to develop compounds that are anti-malarials. One of the problems is with the neglected diseases, and these aren't rare diseases, they're neglected, which means they afflict the poorest of the poor. So even if some of my former employers, the Pfizers of the world, won't even touch some of these because, sadly, there's no money to be made in coming up with an anti-malarial because the patient population simply can't afford the compounds. And so groups, thankfully, like MMV, Medicines for Malaria Venture, step into the gap, into the breach. Whoops. I need to go forward. So these are the neglected tropical diseases that were of particular interest, or the ones that I, in some manner, shape, or form, are working with. So trypanosomiasis, the first one, trypanosomiasis, is sleeping sickness, leishmaniasis, Chagas disease, and malaria. The work I'll be doing today deals with malaria. And it's amazing. Hundreds of thousands of people die a year from malaria. And most incredibly, I was at a meeting once, and one of the most effective mechanisms is mosquito netting. and they can't even get that to those people. They're simply getting bit, and that's it. So, oh, sorry, shouldn't have been so flippant. So when we were working with the partners, now this is 2 employers ago. This is when I was with Synexus, and they had a reason for being involved with these folks. But the idea was they had extant data, and they wanted to know how to do something with it so they could go to these laboratories and say, we want to purchase more compounds. They didn't have limitless funds, and so the idea was we want to be wise in how we choose those compounds. And that's where this work came into play. And so now, what do you do with the external data? Where does it come from? How do you screen it? And then there's another hook to this bit. They had money. They wanted to spend the money on compounds. They didn't want to spend it on computers, software, firmware, or essentially staff. So that's why the driver here are open source tools with open data to build all of these models, actually. And so now it always comes down to, again, the machine learning mantra or the trope. Knowing what I know, how can I predict a future state? Or how can I inform a future decision? So this is my picture. This is the one that makes sense to me when people talk about machine learning and supervised machine learning. So starting in the upper left-hand side, you have some training data. This is the stuff you have and that you know about. It has labels. In, for small molecules, compounds are active, they're inactive, they have a particular IC50. They're active against different targets, off targets. You calculate features for them, and in the area of cheminformatics, that's a blessing and a curse, and I'll get to that as we go to the feature calculation, but that's a big part of the work. And then going forward, and then you build your models, and you somehow populate or distribute those models. As new data comes in, as you calculated features for your extant data, you calculate the same set of features for this new untested data. You then run those features through your model and you get a prediction for every compound. And with luck, or hopefully, you get some sort of enrichment. That's another point to make here. This is a chainsaw, not a scalpel. When people look at compounds to test, they're not going to buy one, test it, and then say you were successful or not because of the activity or lack of activity of that one compound. Rather, it's a library purchase. So they might get thousands of compounds or hundreds of compounds. So close counts. So it's really the sake of enrichment. So it's not necessarily that I need to have these smoke in R squareds or have models that will predict everything, but rather just enough to know that if I was presented with 1,000 compounds, it could only purchase 400, it would be the better 400 out of that 1,000 for the target of interest. So now we start with training data. And when I go into the notebooks, I'll show how you can ingest this data. You can download it and start using it. But ChemBL, which is a branch of EMBL, the European Molecular Biology Laboratory, is a repository for compounds and their activities. And there are literally hundreds of thousands of compounds with associated activities. That's the good part. The challenge is the data is gently curated. So it's left to the depositor to say that, yes, this compound is active, no, it's not. It was active in this screen, not active in that screen. So if you think that you have 4 or 5 different data sets for malaria, actually for any target, the idea of trying to fuse those data sets and say something about them can be something of a challenge. So there are 2 things that are done. One, if you've really got your mojo going, you would build a model against every data set and then use a model from data set 1 to predict data set 2 and see how that works out. Alternatively, instead of going for a regression, you could go for a classification. And you would simply say in every instance, I just have a cutoff. So if it's more than a micromolar, it's inactive. If it's less than a micromolar, I have interest in it. So this is what the 3 data sets look like that I just mentioned. This is going back to about 2010 when there was just a bloom of activity around antimalarials. From GSK, Novartis, that's the N in GNF, and St. Jude's Children's Hospital. And so they tested a number of compounds, and the Venn diagram is to represent which of those compounds were tested in common. One of the nice pressure checks here was in those instances, say the 50 that GSK and St. Jude tested, they generated similar activity profiles. It wasn't spot on, but at least by and large, the screening results had the same trend. And so that made it easier, or at least gave some confidence in being able to combine the data sets and then to go forward to build models. It's chemistry, people. So now here's the cool part. Yes?
So was the goal to find a cheaper medication or a more effective medication?
More effective. What happens is there's always literally the mosquitoes develop, artemisinin is the, is that? I think it's the top one. No, I've forgotten, God. But the current therapies are less and less effective.
So it's evolving.
Yes, it is, yeah. And so really they're still looking for the silver bullet, that one compound that would simply just start attacking the mosquitoes. And then just people, well, actually, I mean, help people, but the idea being that as the mosquito bites, you would have some protection against the consequences. And it's just it mutates, it gets used to the drug therapies that are available today. Today's therapies go back to the 1950s. And then those, and identifying those, it was literally spray and pray. You know, you give people stuff, natural products, and you see what starts to work. And a lot of the earlier compounds were the consequence of visiting the indigenous peoples and saying that there were certain things they would do that seemed to help some people combat malaria. And they just kept going forward. But here, so it's two-pronged. My interest or my effort was to develop a screening library. So there's, MMV has what they're calling a malaria box. It's 400 compounds that's available to anyone to screen. The idea being that'll get your motor running. You could screen those and then go on. I was the computational chemist responsible for identifying the 400 compounds that went into that set. So the whole idea again was we had any number of commercial libraries that we could purchase compounds from. The idea was then, well, what 400 should be ordered and why going forward? So some of it was machine learning for the sake of getting compounds that we hoped were going to be active. A lot of it had to do with similarity searching because, you know, there's always that nagging feeling that we know all of this stuff, but what's happening over here? So let's, you know, it's an 80-20 rule of a sort, and be going into a little bit of that. Yes? Oh, smiles? There's an example going on later. This is a mechanism whereby you can encode information about a three-dimensional molecule in a one-dimensional string. And when I talk about the descriptor calculation, I'll go into a bit more detail, and I actually have a few figures that are just, that'll help us out. And since I had an answer, that was a great question. Thank you for that. Right. So now we have to calculate some features. And this is where, wow, it could be something of a challenge. So the old trope, if you do machine learning, 80% of your time is spent cleaning up your data and getting it ready for the downstream activities. If you go to the UCI, to any of these data repositories, you could download the Boston housing data set. And to a degree, that's locked. There's a fence around it. There are so many features, so many rows, so many columns done, and you can go do what you do. When you start to calculate features for small molecules, the operative word is calculate. And there are instances where for any molecule, it wouldn't be unheard of to calculate hundreds, if not thousands, of descriptors or features. And so you've got to believe that there's redundancy. In some instances, there are probably features being calculated that people calculated them because they could. Here's an interesting one. In playing with this one, so you have a smile string, which is just an alphanumeric string. And in a few slides, I'll show you just what all that means. One investigator simply parsed the string to see if that would have any effect. And it was called Lingo, L-I-N-G-O. So they started in the first instance, the first Lingo string would be C1CC. The second string, 1CC, open paren. And he just did that for every molecule and then used those as kernels so that you could compare and contrast to say what's alike, what's not. And was there some merit to it? And the results didn't stink. You know, it's almost like that alone started to say something about some similarity amongst the molecules. And now we start. So I've got a slide that says this in words later, but I'm just going to let this one go now. When I was in graduate school, I studied DNA intercalants, and I did that computationally. I've never been, well, except as an undergraduate, I've never been in a laboratory, in a wet lab, and the world's a safer place for that. But all the work I did on the computer was with DNA and turculins. And so that was at the interface between biology and computational chemistry or chemistry. And my advisor had a piece of advice. He goes, when you talk to biologists, speak like a chemist. And when you talk to chemists, talk like a biologist. And your erudition will always come through. How many chemists in the room? Oh, okay. I was going to say, we're talking a lot of chemistry here this afternoon here. No. So I can calculate any number of descriptors. And what will happen is a molecule holds, a cord has a benzene ring, a 6-membered ring. And that ring has certain properties. So what will happen is we'll write the code that can deconstruct a SMILE string, and if it recognizes a benzene, then the description or the values for benzene are appended to the set of features for that molecule. And you go on and on recursively. And again, I'll have a worked example a little bit later on. But these go on forever. I mean, there are some instances where there are some laboratories that just continually put out these descriptors. So a part of what I'll be showing you later is how to navigate that morass of numbers to something that might actually be intelligible. CDK is the chemistry development kit. It's a Java tool for working with chemistry. And again, everything that I'll be mentioning is open source and free to use. And again, I'll make a point of it, but if you drop me a line, I can give you the links to download it and such not and what have you. What I'm interested in then is that bottom part, that spreadsheet. So the compound ID is good to sort of, for the bean counters. The PXC50_3D7, that's the activity. And this is basketball, not golf. So bigger scores are better than lower scores. The SMILE string is used to calculate the descriptors. So in this data set, if I go over that way, I want to retain column B. That's my Y value. And then columns D through whatever are my various X's. And so now I've just given scope to the problem. I want to use that feature set against that Y. The other part is, the reason I bring that up is the tools that I'll be showing today, while used for cheminformatics, clearly aren't restricted to cheminformatics. And so the notebooks, and I'll show you why I'm pretty anal retentive in the way that I'm sort of juvenile about how I try to construct the code. You'll get a feel for why I'm a big fan of trying to keep things in data frames or spreadsheets. Yeah?
So back in the lab, what would they measure typically?
Usually inhibition of a molecule against an enzyme, so a lock and key sort of bit. And what will happen is on that binding event, sometimes you get fluorescence. In some instances, there will be an endogenous inhibitor that you would off-compete, and that another molecule would actually, anthropomorphically, muscle its way in. And then you would measure the concentration of what was displaced. And so it's amazing. They can get down to picomolar active compounds, 10 to the minus 12 molar. And they can detect that in the laboratory. Yes. Yes, there are different pathways that are implicated in the malaria event, someone getting sick or whatnot. And then in each of those, there are proteins that you can interdict. Imagine that it's a cascade of events. And if you can, effectively, it's, have you ever seen an 18-wheeler? And what the driver does is put a little block of wood under one of the tires, and that prevents it from rolling backwards. So that's what a drug looks like to a protein. It's just this little bit. It's a pebble in the shoe that just annoys the bejesus out of it, and so it can't do what it really wants to do. And so that's what these small molecules are doing along the way. And actually, that's a really good point. See, now you guys got me talking chemistry. I'll get to the computing. Because what they want to do is go really early on in the cascade. Because effectively, if you think about it that way, you don't want to clean up the spill. You'd rather turn off the faucet. And so that's what they're trying to do when they try to get some of these compounds. Some of it's tough too, just as an aside, because there still needs to be some real basic research done. And this isn't one of the poster children for what do people want to study in labs.
So you said column B is the Y?
That's correct. Yes.
And
No, that's the actual, the XC50, it's an IC50, it's an inhibitory concentration that inhibits 50% of some response. And so this is a negative log 10 of that value. So if something is a micromolar active, that means I need a micromolar of my compound to stop, very simplistically, I need a micromolar of my compound to stop the progression or to stop the activity of a particular protein. And so if I take the negative 10 the negative log of that, so 1 micromolar is 10 to the minus 6th, so it's 6. So 1 micromolar would be 6. 1 micromolar, 500 nanomolar would be 6.3. Something that's a nanomolar would be 8. So it's the bigger the numbers, it has to do with the biological response. And one could calculate similarities, and I'll show you that we do that later on, between the molecules based on the various descriptors, either Euclidean distance or, if you have binaries, Tanimoto distances or Manhattan distances. So. The smiles? Oh, come on. Oh, that's for this study. Yeah, these are either, I'm going to say, I think that's a GSK number. So that's the contributing laboratories identifier for that particular compound. Yeah, it's for ordering, literally, to get more of the same. If you do this as part of your day job, almost invariably, people are going to look at various broad profiles of a set of compounds and say, are they drug-like? And so invariably, they'll look at molecular weights. And there are just some bigger than a bread box sort of questions. You don't want something that's necessarily more than 650 or 700 molecular weight, because if it's too big, it's going to be greasy. It's not going to get anywhere. It won't be absorbed. You can look at hydrogen bond donors and hydrogen bond acceptors. Imagine, if you will, that those are two ends of little magnets almost. If you have too many, your compound's going to stick and never go anywhere. It might be sticking to the wrong thing. If you have too few, it's almost like the Velcro's not working anymore for you. The number of rotatable bonds will speak to how flexible a molecule is. You'd like some flexibility, but not a lot. And so this is just a profile. And you know what it comes down to? I am sure that I could show you folks some code and you'd understand it like that, without any background or any description. This is the sort of picture that when I was doing this, if I showed this to a room full of medicinal chemists, they would just get it. You know, and it's sort of weird. It's one of those things where you generate it so they can see it. It gives a warm and fuzzy that, and looking at these 6 plots, they would say, okay, based on this sort of fingerprinting, the compounds look drug-like, so we're good to go. Now, you want to do some feature engineering. And so in this one instance, for the set of compounds that I looked at, I didn't want any compound that had more than 12 rotatable bonds. If it was too flexible, the entropic consideration of having a fortuitous binding event would just fall through the floor. So it's another one of those, it's just something you look for, and you can limit the number of compounds you have. I also include this side because there are some things you can calculate that are real. If you look at the number of rotatable bonds in a small molecule, and I can show you that when I get a molecule up later, what that means, those are real things. It's not you're using a computer of sorts to do the count, but it makes some sense versus some of the other descriptors that seriously are a very different language. And here, all one looks at are just an activity profile downstairs. So this is the activities, and that's going to be when the compounds have more than 12 rotatable bonds or fewer than 12 rotatable bonds. I do this because what I want to make sure is if I removed any compound that had more than 12 rotatable bonds, that I throw away the magic bullet. What was the activity of the compounds with more than 12 rotatable bonds? Were they active? And if they were, clearly I want to retain them. Same drill. We'll do the same thing with molecular weight. Bigger's bad, or so the belief goes. And again, you can see what the consequence is when I remove those compounds. So that's another thing that people are always concerned about. It's just you don't want to throw the baby out with the bathwater. And so you want, if there's a good reason, you want to retain the compound. So now we actually start doing some code. So this is done in both R and Python, as well as the open source tool for the whole workflows, KNIME. So if you're familiar with it, KNIME has hooks into both R and Python. So it's actually a really comfortable place to work. And again, I've just mentioned 3 tools that are open source. If you've got the bucks for it, KNIME is a whole lot like Pipeline Pilot, and it also has a flavor of orange to it. One of the neat things about KNIME and R and Python is that they all have very active user communities. Have folks used or familiar with any or all of those 3? So R, okay, great. I'm sorry?
How do you spell KNIME?
KNIME. See? Yeah, glad you asked, right? Yeah, and it loads everything. Remember for a while they would say Python, it was batteries included, because when you downloaded you didn't have to do any external loads or anything like that. KNIME is amazing because when you download it, it has all the tools, including all these chemistry tools. So it's a happy place to be. So here's some feature engineering, and this is just some of the code that's being used. And so again, you see just some simple Boolean operations. If it's greater, throw it away. If it's not, keep it. One of the things I do in all of this is I will tag, not toss. So if I had a data set of 1,000 compounds and was asked to process them, and 50 of those had more than 12 rotatable bonds, I would add an indicator column, GT12, greater than 12, and then every compound would be listed as true or false. So what I always want to be able to do is go back to the original data set. And so you'll see that in some of the work I do later. And so as a consequence, if my code starts to get a little bit long, my anal retention really shows through, because towards the end, I keep on adding on to these data frames in R, pandas now with Python as well, but these spreadsheets, so that the original data is always there, and then I just layer on, but hopefully in a sensible way, a sensical way. So now we've got the features, we've got the labels, we know what the activity is, and so now, in this instance, because of mixed data sets, I wanted to do a classification model, so I had to come up with I had to define an endpoint. What's active versus inactive? And here we chose 6.3, so that's 500 nanomolar. Can anybody recognize in a classification problem, there's a glaring concern here? I'm going to call 6.3 my cutoff. So if something is 6.31, it's going to be active. If it's 6.29, it's inactive. And you can imagine that very similar molecules might be looking at each other across that bridge. And so that's one of the confounding issues that I deal with in classification problems. So I'm going to turn it around. If anybody has some sort of a solution, or if you've ever dealt with the same sort of thing where you have, they're not really edge effects, right, because there's something on the other side, but some sort of a partition, and the data right there actually messes up in one sense the answers don't look so pretty, but you want to try to do something with it. I'd be really happy to have a conversation. What I've done is I use a tooth-gapped grin, and so in building some of the models early on, if the results don't look that promising, what I may say is at 6.3, I'll go anything 6.28 or less is inactive, anything 6.32 or more is active, and that little middle piece I just don't do anything with. And see if the model can somehow reflect that. But then again, I'm still stuck with the thing in the middle, but for a classification model, I don't quite know how to, you have to cut it somewhere, and it's always confounding, but we'll let that one be. What I can do within the R world is ask for STR is for a structure of a data frame. And I'll show this in the workbook in just a moment, but the idea is this shows, so compound ID earlier on, that was column A in the spreadsheet. PX50 underscore 3D7 was column B. So I just put it on its end, if you will. And what this lets me do is it looks at each of the variables and gives some feel for their being right or wrong. Case in point, compound ID is an integer. That's not the right class for a compound ID. That should be a string. Even though it's numeric, it doesn't have numeric consequence. It doesn't have a numeric meaning. It's 12 isn't bigger than 15 if it's a compound ID. It may have something to do with when it was synthesized, but nothing more. So you'd want to change that one. And then you can imagine going all the way down. All the others, in fact, are numeric. In R and in Python, there's also a need in some instances, particularly in classification models, to go from continuous variables to factors, rather than so there's true false, or if you have a few states, a number of factors. So we'll be showing that in a moment too. The other part is, remember before I said that I can get hundreds of descriptors, and I need to throw some of these away, or I have to figure out a consequential or a reproducible mechanism for getting rid of redundancy in the data set. So here what I've done is I've calculated the pairwise correlation of all of the descriptors in a data set. And I show it graphically just because it drives the point home that you can see at about, 4 o'clock or so, there are some highly correlated descriptors. And you can imagine, and I'll show this later, actually I'll be using this later, if you're doing some sort of a recursive partitioning or a decision tree, and you had two descriptors that were highly correlated, essentially you'd start spinning your wheels. Because, again, being anthropomorphic, the program or the algorithm would look at your data and say, well, this time I want my left hand to be the deciding to be the split. The next time, the right hand, but they're equal value. They do the same thing to the underlying structures of your data. And so it's good to remove those if you can. And so we do. What we're going to do, so again, just some slides first. See, I'm old school. I'm just going to, another real quick story. A month ago, I started at Solvay Chemicals, and I'm in Bristol, Pennsylvania now. And I was fortunate to get a phone call that started the entire cascade. And in the middle of it all, my wife's going, well, you really might want to think about it because, you're not as young as you used to be. And so they're just, the concerns that you have. And I remember reading a blog, and it started saying how it could be considerably more difficult for older data scientists to get jobs. And they meant 30. And they're talking about people where I have sweatshirts that are 30 years old. And they're talking about people. So why did I get started on that? Thank you. Oh, yeah, so I was going to show the slides first. So then if my notebooks don't work, you're still going to get home. You get to go home with a story. Thank you. How many people use 2x2 slides in any presentation in their career? Bona fide with the carousel. A hand? No? Okay, just me. But, okay, all right. Grease pencils before there were Sharpies. But anyway, so when, and here in this slide, what the program does is it looks at the pairwise correlation of every pair of descriptors. And the user, I, set a threshold, and it's usually somewhere around 75 to 85%. For every pair of descriptors that are that close together, another step is taken, and it's determined which of those descriptors by itself is most highly correlated to the outcome. And that's the descriptor that's retained. Now, there's a real important reason for this, and this is one of my bugaboos. Everything that I show you will be absolutely reproducible. If you follow the steps, if you execute the notebooks that I will share with the data that I will share, you're going to get the same answer. I guarantee it. Because in too many times I've read papers where people say, we removed highly correlated variables. That's all they say. And so at that very point in time, they've broken the chain. There's no right way to reproduce their work because how did they decide which of the pair to toss? And so it's one of those things, if there's anything that you'll get to take home from all of this, for as spaghetti some of the code may look, if you run it step by step, you will get the same answers, and I guarantee. Okay, so now we go into the machine learning algorithms. And here's the code. And again, this is in a notebook, but it's on a slide. So it's pretty easy inside R to start building up some of these models. So you can see in the first instance, the random forest, that word is what invokes the model. So rf.model is the model itself. And what it's doing is it has a formula. So the tilde inside R essentially, it defines the formula. To the left of the tilde is your y value. To the right are your independent variables. And if you use a dot, a period, it means everything in your data frame except what's on the left-hand side. So in the first instance, what it's saying is, I need this as a factor, again, because I'm doing a recursive partitioning, a random forest, and I'm doing it for classification. So it needs to be a factor. But my endpoint is active or inactive, and that's going to be dependent on every other variable in my data set. So that spreadsheet that I had the PXC50 and then descriptors. That's all I need, and I can start building some models. And over on the right-hand side, you see how good the model is or not. Are folks familiar with confusion matrices? Okay, because they're aptly named. Let me see if this is. There's not a. Whoops. Not a clue. I don't think this is it? On? As I blind myself. Oh yeah. Is it? I think I can see it here. I think yeah. Okay. But the idea is, if you look down, the confusion matrices, what they say is it counts the number of true positives, true negatives, false positives, false negatives. So if you look into the table itself where it says reference and then prediction, I want the upper left-hand corner and the lower right-hand corner to be really big numbers. And I'd like the lower left hand and the upper right hand to be relatively small. The true positives are in the upper left-hand corner. The compound is active, and you predicted it to be active. The true negatives are in the lower right-hand corner. The compound is inactive, and you predicted it to be inactive. I think I'm going to get this right. Are there statisticians in the room? It doesn't matter. Just believe what I say. The class 1 errors are the upper right hand, where you are predicting the compound to be active and it is inactive. So you have a false positive. Your type 2 errors are in the lower left-hand corner, where you are predicting a compound to be inactive, but it is positive. So you've got a false positive, false negative. Right? Did I say that? Well, what I always walk away with is upper left and lower right big numbers, lower left, upper right small numbers, and you start to walk away with a good result. Then it talks about accuracy, sensitivity, specificity. Those address how many times do you identify a real compound as being active when it is active versus all the background, or how many times do you properly identify the inactive compounds versus all the background. My takeaway is always the kappa value, because it allows for chance assignments. Suppose I had 100 compounds, 95 were inactive, and my model said your compounds are always inactive. I'm right 95% of the time, and the model's useless, because it's never going to identify an active compound. The kappa variable mathematically accounts for that sort of chance error. So you can't just always say that the greatest number of occurrences is what drives your model. There's no absolute for how to interpret kappa, but, right, so I'll contradict myself. If it's somewhere around 0.5 or so, you have a model that you could probably use. If it's upwards of about 0.6, 0.65, you've got a model you can publish. Again, these were some initial efforts. So somewhere around 0.4, edging up that way, but there's something you may want to do. In my hands, I use kappa as my target function. So if I start to compare any number of machine learning models using any number of descriptors or any number of methodologies, kappa is that single value that I look for to progress my work. So the bigger the kappa, that's the one that I'll carry forward. It's proportional. So I've got a slide for that too. What I'll do is if your entire data set is, if there were 40% actives and 60% inactives in your entire data set, those proportions are carried forward in your training set and in your test set. So it's not random. It's random within those tranches.
But if your data is
Mm-hmm.
like in this case, we have a lot more inactive, and your model, by default, predicts to be inactive.
Yes. Well, two different things one can do. One practice is to balance your training set. And so literally don't look at all of the inactives and then do a leave one out cross correlation or something, because now you're making your data set smaller, but you need to account for the fact that it's smaller. You know, you can't quite do as robust an analysis as you once could. So some n-fold cross-validation could be used to leave one out cross-validation with a smaller data set. Again, holding out both actives and maybe many inactives, but a few actives, and seeing how well you predict against your holdout sets. Invariably, there are unbalanced data sets. You know, it's descriptors. You can do a Box-Cox transformation. You can do PCAs. You can do all kinds of stuff with descriptors to somehow try to make them better behaved, but you can't do anything with the real data. You know, if it's skewed, and you just try to see what you can do. So, but I've seen people, they try to balance their training set, and literally, they don't throw away the data, they just don't consider it all in the model construction. And then the other piece, to further confound it, is what I may want to do then is say, if I have, for every active compound, I want to try to find a compound that's similar but inactive, and let me build up a data set that way. Because if I can distinguish between actives and inactives from near neighbors, from things that look a whole lot alike, I've got a really strong model. Because if there's something that is inactive and doesn't look anything like my active compounds, I'm not sure I'm learning anything. And so you could sort of run it out that way. So I'm one of those people, I don't think that there's ever going to be automated machine learning. Because there's too much you bring to the table when you get the data. Yeah.
Is the same for when you have like a binary prediction versus the variable the severity of the cost? Like active versus inactive. Is the confusion matrix the same?
The confusion matrix, so I do an R squared, or I would do some sort of a line fit if it was a regression. So the confusion matrix, the confusion matrix would be for a classification model. Yeah, and a good question though, because you can also do it if you have 3 or 4 stages. The grid just gets bigger and bigger. And again, it's always the main diagonal from top left to bottom right that are the true whatevers. Okay, I'm just going to blow through the next couple to get up to the workbooks. So then a support vector machine. This is going to be theory light, and I'm in a moment practice heavy, so I'm not going to go into the details of why you would use one or the other. What I try to do is keep all of them in my arsenal. So again, you can see the SVM model is nothing more than the training set, the X's and the Y's, and it can start to build a model for yourself. And so now in this one, we're doing not quite so well. The kappa fell off a little bit. I should say all of these are using the same set of descriptors. So this is really a test of the methodologies against the same set of descriptors. And then an AdaBoost. And the kappa gets a little bit better. Here, the thing to point out is with all of these models, so I'm using a package called CARET, C-A-R-E-T, that's available through CRAN, the Comprehensive R Archive Network. And CARET is for classification and regression training. And what one can do, and you can see in the control, is a number of these machine learning methodologies have parameters themselves. So the variables you choose are one body of parameters, or the variables are one set of variables. Your training parameters are another set of variables. So arguably, I could hold the same set of descriptors, the same methodology here in AdaBoost, and then change the control parameters and get different answers. So really, it should keep on peeling back the layers of the onion, and there's something else to do yet. AdaBoost is one of the, as boosting does, is simplistically, because that's the way that I understand it, it will run a set of a set of instances, and on those that it predicted poorly, it goes back and essentially doubles down to try to predict those properly in its next iteration. And that's the boosting bit. So it essentially says, if I change a thing or two, can I capture more of my wrong answers in the first iteration without generating more wrong answers going forward? Again, if anybody wants to talk deeper, and I'd be happy to, afterwards maybe or what have you. And so now to introduce new data, there's always a question of, can I model a new data set? Is it within the realm? Because you can imagine, going back to the earlier example, had I built a model on molecules that had less than 12 rotatable bonds and a molecular weight less than 600, and you presented me with a number of macromolecules or macrocycles that had more than 12 rotatable bonds and molecular weight of 800, I might simply be looking at a body of compounds that are not a part of my training set. And then in some instances, proper assignations of activity might be spurious rather than being something that the model's really bringing to the table. So my go-to, and this is only me, is I just do a PCA. So, and I'll show you in a moment how that goes forward. For any data set, once I've just, for my training set, this is the gold for me. Once I have a training set and its descriptors, I'll then use those descriptors or whatever I've done to machinate those descriptors and run a PCA. I then apply that same schema to my new data. So if I found that, A and B were highly correlated and I threw away B, and my new data set comes in, I throw away B without even looking at it because that was not a part of my training set. I'm going to throw away that variable. Then I'll do the PCA and I'll see how well things fit one to the other. And so essentially, and the gestalt of it here tells me that the three data sets are coincident. So if I build a model off of any one, I ought to be able to predict the consequences of the other two. So that's the whole idea of doing the PCA. And plus, if you're in industry, it doesn't have to have pretty pictures it doesn't hurt to have pretty pictures every now and again, because then people are going, oh, that's what he does. In this instance, yes. And just for visualization. So one, as an aside, one really cool thing is, if you're not familiar with it, it's called GGOBI, G-G-O-B-I. And it's also available through R, and it does projection pursuit. And it is just wickedly cool, because what it essentially does is it says, take your n-dimensional data, 3, 4, 5, 6 dimension, doesn't matter. And particularly if you have a classification problem, it will project onto 2 dimensions, and it continues to rotate in your hyperspace until your actives are separate from your inactives, or until literally the white space between them in the projection is a maximum. And it does it while you're watching it. It's mesmerizing. But it's just very interesting, because it's essentially saying that there may be a splitting plane. So it's maybe it's got a flavor of a support vector machine too, but what it's saying is there may be a hyperplane that will separate your data, and it tries to find it. So it's GGOBI. It has its own user interface, and it's also available within R, so you can send data there and come back with it. And now, good, okay. So now the idea here was I'm sorry, is there a question? Oh, okay. What I wanted to do here is predict the activities of the St. Jude data set. I use that as my true test set. So now, how can I use this going forward? And I'm finally getting to the end of these slides. There's a secret sauce inside R that works, and it works for me. In everything that I've done, in all of the work that I've shown you, R provides a mechanism where I can save, and I'll show this in greater detail in the notebook, a transformation. So did I do anything to the data? So did I change an IC50 to a PIC50? Or actually with the descriptors, did I mean center all of my descriptors? I can save that transformation. Or were there near zero variables? I'll describe that in a moment. Or things that are highly correlated. I can save all of that, and then I can transfer that model and use it anywhere else. And as soon as I apply the model, if any new data set has at a minimum all of the features that the model learning data had, then we can go forward. So if I come in with a new data set and have additional features, that's okay. I'm simply not going to use them. But to use the tools here, if I have features named A, B, C, D, and F, any new data set with A, B, C, D, and F can be handled through this mechanism. And again, the notebook will show this in greater detail. But to me, I don't know why, this is just wickedly cool, because then I could start sharing models with folks by giving them these little bits and pieces, and they could start using the models. And when you calculate activities through these models, it's blazingly fast. So the models have been built, they've been verified, you're happy with them, and now you can start using them. And so that was the idea of all of the work, actually. And so then I can just bring in a predicted model. And so with the, so now in the external test sets, the SVM didn't do so well. You can see that the lower left-hand corner, that's too big of a number, if you will. That's off of the same data, the St. Jude data set. It looks like AdaBoost did a little marginally better, and Random Forest may be best of all. But again, not great, but a little bit better of those three. And this is for demonstration purposes. One of the things that I didn't do in pulling this one together is when you hammer down into the data sets is the control variables change. There's a bunch of stuff you can change. And just to leave it like that. The other part that's kind of cool with an R at least is if your penultimate command is just to see what the system is, this will tell you all of the software that I used and the version of that software. So it's reproducible down to the actual code that you're running. Again, this goes back a bit. So this is before things like Docker or other wrappers around a virtual machine, if you will. But for oldies like me, it's always kind of neat I like working with paper. It's kind of neat to see what version of the software is running and knowing that you could reproduce it yourself. So now, just a few more. People will talk about R being predominant or their go-to, or Python being their go-to. I don't see it different. I really don't. I don't advocate one over the other. I think they both have strengths in different places, and I see them being used together. So the work that I'll be showing going forward, I just bounce between the two. So there's no, I don't use R until it doesn't work and then use Python. There are just some things that work better in Python. 2D representations of molecules, I think, is far stronger in Python than in R. So I'll just send over a file with smile strings and represent my molecules and just bounce back and forth like that. There are different models of the whole thing of machine learning. In the upper left-hand corner, this is the CRISP DM, the cross-industry model for machine learning. Down in the lower left-hand corner, if you've read or looked at the books within the R universe, this is the tidyverse, the R for data science view of how you actually start to develop models. Again, you've seen this one because this is my fave. I get this one most of all. For this one, what I'm going to show in the notebooks is I'm a big fan of Legos. And it was really good because I have a son, he's 20 now, but when he was younger, he was my beard, right? So I was like, Tim, we got to go to the Lego store because you need something. And, but anyway, there are a bunch. And I marvel at that, and this is what I'm going to be showing. So the notebooks that I'll be showing are the Lego bricks in the upper left-hand side. They're going to be really granular. It's going to talk about some of the machine learning steps that I've already discussed. They're not going to be the Millennium Falcon, something that's amazingly wrapped into an API to be shared. Rather, what I want to do is show you how I do what I do when. The other thing I think that's really cool about Legos is since there have been Legos, they have all looked the same. And you can fit a Lego brick today with one you got 20 years ago. And they have that sort of interoperability. So in the work that I'll be showing, I try to write my code so that you start with a spreadsheet, a data frame, you do something to it, and you populate or you add to that data frame. Because I'm a real dullard, and one of the reasons I like that is if I can do that with somebody else's code, I always feel that if I can make my data look like your data frame, then I can use your code, because it's going to be doing the same things. And so that's what I try to carry forward. So you'll see a lot of flavors of that. And this is just a whole other piece of it. So the idea is I start with some sort of a data frame, some sort of a spreadsheet, put it through an exercise, put it through a brick, and then I somehow add to that spreadsheet. These are the commands that you're going to see again in the notebook. These are the commands to download the data for, in the first instance, GFK, then Novartis, and the HTS is the St. Jude data. So all the stuff I was talking about before, this is the real deal. And this is how you can download the data. This is the actual code. And so I bring that up because having done that, once I've read in the data, and again, you'll see this in the notebook, these are the commands to read in the data. What I'll do in a notebook, what I'll do with anyone I work with, is I'll then show something about what I've read in, because now I get it. Because people talk about it's indexed or it's use a colon to say what's going on. I like that bottom part where I know that my data has 4 columns. It's got a compound ID and activity, a SMILES string, and a data source. And that's true for all 3 data sets. So now I can combine them, but I get it. And then if I have to do any slicing, I could do it off of indices. So this is 1:4 if you want to look at it that way. But to me, more sensibly, I have a name for what I want to try to call out of a data set. So I'm trying to make excuses for how spaghetti my code's going to look in a minute. So this is the bit. The source, in the GitHub, you will see the data sources for GSK, Novartis, and St. Jude, and they'll be in a data folder. So if you read from that data folder, you will generate that spreadsheet. So up on top is data. Is this going to... You guys seeing this? Oh, good, great. Okay, so this is the data. This is the brick. This is the expanded Excel spreadsheet. And the reason I say that, too, is if you have other kinds of data, it doesn't have to be chemical data. You can still do things with it. You can move forward with the code that I'll be sharing. And to be truly anal retentive, see, I made it look like spreadsheets. I put little double underlines and stuff like that. So, and again, I use CDK for a lot of the descriptor calculation. It's the chemistry development kit. It's a Java program. It has an API wrapper so that you can call it directly from R and just go forward. These are just a flavor for some of the descriptors. Now, back in the day, this was eye candy for me. I'd look at these and say, yeah, I'll calculate some more. These are the sorts of things that I can go after. I'll use some of the words, but just to introduce you to a few of these. So a log P speaks to a molecule's greasiness. And if you're developing compounds, you don't want them overly greasy because then they won't cross a blood-brain barrier. And that's a problem. You can have topological that speak to, again, they're only using those SMILES strings. So they're essentially guesstimating how spheroidal a molecule may look. Is it longer than it is wide, if that can make any sense? But you can see that in some molecules. The TPSA, that's total polar surface area. So every time it sees an oxygen or a nitrogen, it brings a number into play, and it starts to build up features and descriptors that way. So that's what I'm saying, too. This is the other part where it gets sort of confounding, because there's no way back from these. If I told you that a molecule has a petagene of 12, that means nothing. There's no way to reverse engineer that. So this is really, you calculate the descriptors and then see what the machine learning exercises can do in helping you find more active compounds. So it's good and bad. If you work with them enough, operationally what one would do is you take a bunch of molecules you're interested in, calculate some of the descriptors, and again, do the breadbox thing. Is it bigger or smaller than a breadbox, and what does that mean for my molecules? So once again, I just wanted to pass on what my advisor said. So well, there's one other chemist in the room, but this is a chemistry talk. Now onto the SMILES string. Does anybody know what molecule that is? Yes. All right. So now, and I built it up just because I didn't know how good the pointer was going to be. That is a SMILES string that uniquely identifies caffeine. So the first carbon in the string is the carbon that I'm pointing to. And then you can almost imagine, I start walking around the ring. So I'm going to go, and I'm going to go in a clockwise position. And capitalization counts. Capital letters are aliphatic. Lowercase are aromatic. So accepted for fact. And then, in truth, generating descriptors or talking about that area of cheminformatics would be a course unto itself. So we'll just throw caution to the wind and just accept what I say, right? So the first lowercase nitrogen is the nitrogen that has a line drawn to that first carbon. Because it's in a ring and aromatic, it's a lowercase n and it has a number 1, meaning a ring starts there. Then if you go around clockwise, you see another carbon, then a nitrogen, then a carbon, but that carbon's at a bridgehead, so it is responsible for starting a second ring. That's why you have C2. And I'll just, I've got some more red lines. This might, hopefully. So then the double bond O is a carbonyl, that's the oxygen. And then you can see some of the ring bonding. So that first carbon is the first in the ring. The C2 is the carbon that's kind of in the 12 o'clock position, and it opens up a second ring. And the very last carbon has a 1 and a 2, meaning that carbon closes ring 1 and closes ring 2. And it's from these that we calculate descriptors. So this is an amide, a nitrogen bond to a carbon doubly bonded to an oxygen. That means something. So from the SMILES string, when I see an N, a C, a double bond O, that amide means that I have values for particular features that I can tie back to a physical event. And that's how you start to build up the feature space. Caffeine is derived from a purine ring. And there are properties associated with purine. So if I had to do some modeling around caffeine, some of the descriptors would admit to purine. And then there's the SMILE string for purine. And you almost, if you start staring at both of those, you can kind of see where the pendant groups come from, the methyls, the CH3s, and the carbonyl oxygens. We're finally there, folks. This one? Whoops, sorry. It's not. Yes. Yeah, it's really wild. They use @sands, forward slashes, and backslashes all mean something. Triple bonds are the pound sign. Just as an aside, now they have INCHI strings, INCHI. So you can have a SMILE string, you can have a canonical SMILE string. So you could represent a molecule different ways. So if I traversed it clockwise and you traversed it counterclockwise, we would both represent caffeine, but the strings wouldn't look the same. So what one does is before doing anything, you canonicalize your SMILE strings. That means you apply some rules as to how you go through the ring. INCHI strings, the INCHI, are the International Union of Pure and Applied Chemistry came up with a set of rules. They are invariant. So every molecule has one INCHI string. And so now what people... Yes, exactly, that's right. And so what people are starting to do now is essentially reverse engineer. So they know how to calculate things for SMILE strings. Now they're doing the same for INCHI strings. It's actually kind of cool. So this is an R notebook. So I used RStudio, and what I'm just going to walk you through is the steps that I've executed to build a machine learning model. And it will be every step, because I want to try to highlight some of the things that are necessary to build up models where they make some sense. So hoping, it won't be. So you always have to load libraries within R. So here we're going for rpart for some of the recursive partitioning we'll be doing, as well as partykit. This is something that will help us draw some representations of decision trees as we go forward. So you remember when I took German in college, so alles is the full data set, but that's everything. And so when I do that, the DIM in R means I want to get a dimension of the data frame. So this tells me that I have some 19,000 rows and 62 columns. And I will always do that. So if you see this code, or if we start a communication and I share any other code, you're always going to see this sort of thing, because to me this is a pressure check. As an aside, for as long as I was working with chemical companies, and still today, I've never met a colleague whose data was as robust or as clean as she or he thought it was. So invariably, they'll say, we have a really big data set, and I'll do this little game, get a dimension of it, and it's 120 rows. And I go, no, wait, we have more. No, you don't. And so this is a little bit of a pressure, just to let us know what's going on. The next one down, the command complete cases, identifies any missing values in any position of any row. Because invariably, there might just be some blanks. And in fact, there were 4 compounds that had no activity reported. So they're of no use in any part of my studies, so they're removed. And I do that with the NA-omit command. So they're gone. After having done that, not surprisingly, I've gone from 260 to 256. I've lost those 4 molecules. You remember before the STR for structure. Now here's where, folks, I invite you down into the weeds. I promise there's licorice when all this is done. But just to go through every step. So here, as I start looking at this, the source, I go up here. The compound ID is a factor. That doesn't make any sense. The compound ID is a string, a character. So I'm going to need to change that. The P activity is a number, that's good. Smiles is a factor, again, makes no sense. It's a smiles string. So I'm going to need to change that. And then it looks like numbers, integers, that's okay. Everything else is clean. So here you can see, and this is the command, I force the compound ID to be treated as a character. And that's all R does, and it does it for me well. Similarly, the smile string is now a character. And I just quickly check again. I check on the class, that's the R command, and I see that they are now both character strings. This is going to be important because a lot of times in R particularly, Python as well, you get a lot of the gotchas where you're, where you're moving along and a program won't run because it doesn't have the right class. And a number of the models within R require factors, or they'll call them levels, but those are factors. And so I always try to keep my data clean going in so the modeling is going to work out for me. What I've done here is just a density plot of the activities reported for the compounds in each of the 3 data sets. And again, this is just another test, but you'll notice something interesting just at this. If I sort of skew my eyes, the Novartis and the GSK data kind of the same distribution. There's a lot more of the GSK data, so you see that spike, but they're a little bit broader. The St. Jude has a lot of inactive compounds. So you remember earlier on, part of my activities was to try to predict the St. Jude data set, but there are a lot of inactives. So to an earlier point, you might want something a little more balanced, or you might want to take away some of the inactives on the St. Jude bit, just because they might be confounding as you go forward. Next down, again, this is just looking at the activities and their distributions. This is done using Plotly, which is a standalone JavaScript tool, but it can be incorporated into R. And now this is running inside of an HTML, and you can interact with your data. So this shows you the box and whisker. You can see your first quartile, your median, your third quartile, what have you. You can do things. You can zoom. So, whoops, I did that. There we go. So if I only wanted to look at one data set, I could. I think this is kind of sharp because this is inside an HTML. And so if I mailed this to you, you could be doing the same thing. So it's fully JavaScripted. And then let's see. Reset axes. So then it comes back to itself. And this is kind of a cool one. I can also, at any point, generate a PNG of the visualization that I'm looking at. Maybe not so interesting here, but you can also do 3-dimensional rotations and capture them. So if you had a particular projection that you wanted to share with friends and colleagues, you'd be able to do that in this environment. We don't need that. So now I'm going to build training sets and test sets. And I'm just going to move along a little bit to show some of what can be done here. So here's an instance where I've calculated the number of actives and inactives for each of the 3 data sets, and I'd like to report that to someone. But right now I have, it's not quite the format I want. So it's an easy transformation within R to spread the data. So I'm pivoting the table. But now I know that in the Novartis data set I have about 2,000 actives and 2,300 inactives. And so again, just a single command and you can very quickly massage your data into something that's attractive and useful. The other bit is I've done a bit for each of the data sets. They're purposely built so that as I start to merge them, I know that they have all of the same column headings so that the merges will be clean. And that's shown when I know this to be true, and then I do a structure on each of them. So now that's just, we'll rip through that one. And again, the same. Like I said, I told you, it's going to be little bits. But again, there's a part of me that's driven by this because now as I work through this at every step, I can see what I have or haven't done to a data set, which will become important as we go down to some of the modeling. And here, again, I'm sorry, I literally printed out everything. So now, when the question came up before, I'm creating data partitions. And what this will do is it takes my endpoint data, so my activity data, and it's going to retain 80% of that data for my training set, and the remaining 20% will be my test set. And then when I go down a bit, and it does that by identifying indices, and so then I can slice based on the indices. When I go down here, this is a comparison of the activities, the densities now, of the training set and the test set. So this is a clear data partition. So I like this bit because now what I am sure of is as I go forward, I have the same depth and breadth of activities in my training set as I do in my test set. So there's no skewness there. That's kind of a cool thing. Now the other thing that I want to do is also be cognizant of the range of the various variables. So here I'm just looking at 3 of them. I'm looking at a log p-value, surface area, that's the VSA, and a molecular, the SMR is molar refractivity, that's sort of the size of the molecule. If I look here, let me pull back a bit. When I'm looking for our values that are near zero variance. So in any data set, it may happen that if I have 100 values, they're all uniquely zero. Well, I can take that out of my data stream. I don't need that one anymore. Or it may be that 2 of them have a value, the other 98 are zeros. And those I'll also take out. And it makes sense. It seems kind of weird, but you could remove them because arguably, if you had so small a representation of a value and everything else was either 0 or unit valued, when you do your training and test splits, there's too good a chance that the variables that have a value will always be only in your training set. And so then that could potentially skew models going forward. So then, so near zero variance descriptors, the descriptors are removed, not the compounds, but the descriptors. And as you can see here, this is what happened. So in 2 instances, they were, uniquely 0 throughout the entire data set. And in another, there are some high values, but everything else was 0. Next up is I'd like to look at, I want to mean and center my data. And here's the reason. In the first instance, I can look at a log P, a molecular weight, and a fractional surface area. And if I look at their dynamic ranges, you can see that the molecular weight is huge. The numbers are big. Not so the fractional surface area or the log P. So what's going to happen? If I build any sort of a model, No, it's done off of the connectivity, the topology of the molecule that's built from the SMILE string. Yeah, these are. I admit a lot of these descriptors are an elephant skating on thin ice, and it's getting hot outside. You know that it's hard to really draw a straight line saying that makes some sort of sense. But thank you. Actually, that reminds everything all of my work is based on calculating descriptors from 2-dimensional representations. So a blessing and a curse, actually. So what would happen then in this instance is if the molecular weight changes a bit, that's going to be the tail wagging the dog. If you see any influence, well, I could get a molecular weight change of 10 units on the dynamic range isn't that much, but it would actually wipe out any of the other features that I'm dealing with. So what I do is I mean center the data, and now I get a representation like this. It's a little bit harder to reverse engineer, but as I go forward in modeling, changes in molecular weight now have the same influence as changes in log P, as changes in molar refractivity.
Do you create the column or do you just change the values?
I've done both. But in any of the models, I always only use the new column. So, yeah. And see, that's the other thing. That also raises a good point. For the sake of reproducibility, up here, you'll see that, and I've got a, it'll come up, I'm sorry, I'll do it on the near zero variance. Sorry about that. So here you see that I calculate the near zero variance of my training variables and save it in a value NZV. So this is why it's important that my mu data or my test data have the same columns as my training data, because down here I'm removing those columns by the same formula or the same mechanism every time I remove those columns. So essentially, in any data set, I'm removing columns 31, 36, and 42. And here, for the near zero variance, they are removed. Yeah, I can't. You could replace them, or you could, arguably, you could sort of have a ghost data set right next to it if that was, but I've removed them, or they're removed here. We've seen this before. So this, again, is the correlation. And what one can do is find a correlation. Here the cutoff is 85% or 0.85. So when the correlation is greater than 0.85, one of the two is taken away. Which one? The one that is less aligned with the activity you're looking to model. And so here are all of the values that are being removed. Interestingly, knowing these, there are any number of them that are molecular weight or molecular size related. And again, it's because the calculations are almost redundant in the way the features are determined. So here's the original, and here's the final. Again, having removed the highly correlated variables. And so now at this point, we can actually do some modeling, right? And finally, we're ready to build some models. The first one's going to be a very simple recursive partitioning, just a classification tree, that's all. And here is where you do a split. You look at each of the variables, and you determine if I split a particular variable on a particular value of that variable, can I start to sequester actives from inactives? And this runs very fast, but it also builds rules, which is kind of cool. And in Weka, another machine learning package, if you haven't heard that one, WEKA, again, open source, this one's Java-based, they use a one rule. And one of their ongoing practices is for any data set to always try one rule, because in some instances you may be surprised how frequently one descriptor might be very closely correlated with your outcome, with your activities. So again, my age and retention is showing through. Here's everything that's calculated about the model. Here it is in words, and in a moment I'll show it in a picture. So the words are saying for my primary split, if my molecular refractivity, one of its components, is less than 1.4, and I go to the right in a tree, I improve a Gini index. That means I start to improve the purity of the branch. I'm not going to go into all this sort of thing, but when you do the recursive partitionings, random forest, what you're trying to do is generate splits so that your leaves, not your branches, but your leaves, are relatively pure in actives or inactives. And let's just cut to the chase. There's the picture that one generates from this first exercise. And so I'll read it. So this says, when that molecular refractivity, true, you read to the right if you match the question, and false, you go to the left. So if this value was greater than 1.4, I would traverse here and would find that if another chi value, and again, this is a topological index, was greater than, remember, positive this way, was greater than negative 0.9, let's say, I come down to an active node, because inside this node, there are 209 actives and 57 inactives. See, it's not particularly pure, but it's enriched in actives. And if I look at any of the other nodes, Here, 291 actives, 477 inactives, so a democratic scoring makes that an inactive node. Similarly here, the inactives trump the actives. But you can get this sort of picture. Cool thing about the recursive partitioning is you get this tree. And there are any number of ways of embellishing what it looks like, but you'll always get this tree. You can also calculate variable importance. And so now this is kind of neat. And again, what I also like about the notebooks is I'm going to step through this table in the HTML, and it's a way that you could share it with your colleagues. You note that there are values here. Their absolute value doesn't mean much. Their relative value does. So what this tells me is a log p-value was the most important variable in determining activity of this set of compounds, so how greasy something is. Interestingly, you'll see that the first few have numbers, and then none of the others do. So what this tells me is there were just a few features that the program split on to try to separate active from inactive. So if at any point I needed to do further feature engineering, this would be a great place to start. If the variable is never split on, you get to lose it. So that's just another thing that you can do there. Now I'm going to build a random forest. And again, I can get a bit of a confusion table down here. The formula itself we saw earlier really is real straightforward. So again, I'm using the formula invocation. So the endpoint is a function of all of the features. And I can determine how many trees it's meant to cycle through. So if you increase the number of trees, it takes longer to run, but arguably you get a better result. So I get a bit of a confusion matrix down here. But again, so it's not that bad, but you still see some pretty big numbers in the false positives and the false negatives. And I, again, can identify the variable importance. And I can do that graphically in this instance. And so here the log p is still pretty big, but this chi4, another topological index, is also important. So the shape somehow of the molecules is playing a pretty big role. Here I'm going to go and this is the prediction schema that's used by Caret, which is really pretty cool. So the formula, the format is always predict, the word predict, an open parenthesis, your model name, followed by a data frame of the descriptors of your unknown entities. And then it will predict the desired outcome. And it's just that easy everywhere and all the time. So here, we're looking at a confusion matrix for this random forest. And you can see this one is really poor. So it's probably a consequence of the descriptors, but I've got a very poor kappa. And you can see I have far many more false than true positives. So not a good model. But again, full disclosure, just to show you folks everything. The next one down, I'm doing a linear model. And when you do that within R, you get the variable importance by way of their p-values. So if you were to build up the linear equation, this would say that the number of rotatable bonds is important. One of the components of a log p is important. A few others a little bit less so. Again, more stars is a better p-value for the significance of being included. And then you can get an adjusted R squared, which is really rank. When you plot this out, what happens is it does a good job for inactive compounds. Again, this is a linear model now, so it's regression, not classification. But it's doing nothing for the particularly active compounds. So there are some features of the active compounds that aren't being captured by the descriptor set. What I should mention is you're seeing all the warts here. To come up with good models is a laborious process because there are a lot of knobs and dials to turn and play with. But one just, you just keep going forward in trying to do so. In the next instance, let's see, I'll just, for the sake of time, let's see, I want to get down to the bottom where I actually had some relatively nice results using a method known as k nearest neighbors. So what you do is for every instance, you calculate by some mechanism its n nearest neighbors. Here I used 9. And then what you say is, if 5 or more of those near neighbors are active, my new compound will be predictive active. If 5 or more of the nearest neighbors are inactive, my new compound will be declared inactive. And you can change how many new neighbors you want to include. For simplicity's sake, you choose an odd number, so there's always a winner. And you just keep on cycling through. So 9 ended up being the best for me. Upstairs, there it is, the k equals 9. I started at 3, 5, 7, 9, went up to 15. 9 was just the best player by the kappa value. And so you get that sort of thing going on. 2 more real quick. This is another one. This is a solubility calculation. And again, I'll share all of these notebooks so that you'll be able to do this sort of thing as well. For linear models, what one will often do, to a question that you had about the confusion matrix, was, so here's a continuous or a linear model, and what I'll do is I'll plot the predicted versus the observed. And so ideally, it's on the 45 degree, right, because you're nailing everything. So you can see that there is some noise here. What one then plots are the residuals versus the predicted. And you really hope that that's a random distribution of points, because then there's no underlying structure. You haven't missed anything yet. And so again, you'll see in these notebooks how you can calculate both of these. And I think there's one other here. And just here, what you can also do, and that's this idea of the grid. If you are building any models inside Caret and there is a lambda, if there's something that you can set, you can identify a sequence of those values. So you can algorithmically walk through all of the dials. You can just let it run overnight and come back, and it actually identifies the best model, usually by an R-squared metric. So if, say for the number of trees, I could have built a grid that said n tree equals 3, 5, 7, 9, 11, 13, 15, just let it run and come back to the result. I don't have to do each one in turn. That was just the other piece to show here. And then the other one, and I won't just in words, but again, I'll be happy to share this with you, is, there we go. This is just to show all the work has been repeated with Jupyter notebooks. I've been working with R longer. I just wanted to know a little bit more about Python. So they're mirror images of one another. But like I mentioned, the graphics, molecular graphics inside Python, I think is a little bit stronger. So you can actually see some structures. And here, these were compounds that were clustered algorithmically. And if you look at those, even if you're not a practicing medicinal chemist, you'd say, yeah, I get it. You know, there's a particular gestalt to all of those, and there is a sameness to them. So almost. Thank you ever so much for hanging for the last hour and a half on a Friday afternoon. So I really do appreciate that. I do have liquid if anybody wants to come up for a piece. And if you will, ping me or let me know, and I can certainly send you all of this before it gets all to GitHub, because I'll be sitting down with my manager on Monday and just saying, well, you know, the cat's out of the bag, so it really doesn't make a whole lot of sense not to put it somewhere. But this will all be available here. And it's extraordinarily anemic now. It only has the data sets. But you'll see that growing in days to come. So that'll have, what I've showed you, it'll have the R notebooks, the Jupyter notebooks, and a shiny application, a graphical interface that'll let